Good afternoon and welcome to MycoTalks. MycoTalks is hosted by the MRC Centre for Medical Mycology at the University of Exeter. My name is Alex Brand and I'm one of the PIs at the MRC Centre. The co-chair today is Professor Judy Berman from Tel Aviv University, Israel. Today our speakers are Professor David Denning from the University of Manchester and Professor Anita Sil from the University of California, San Francisco. MycoTalks are organised by the committee shown at the bottom of this slide with valuable assistance in running the event from the MRC Centre staff. MycoTalks are held at 1600 hours UK time on the last Thursday of every month, usually, not this month or next month. They were started by the MRC Centre back in July 2020 to bring together the medical mycology community to hear about great science during the lack of face-to-face conferences due to the pandemic. Even though we're now emerging from the pandemic, MycoTalks attracts an audience of around 300 scientists around the world, with many more viewings of the recording on YouTube later on. Apart from great science, the philosophy of MycoTalks is to promote diversity in our field in terms of career stage, gender, geographical location, and research field. And the committee is always happy to receive speaker suggestions for our future programme. The format is two 30-minute talks on different aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will be seen only by the moderators. It's important to state the name of the speaker you wish to address. The chairs will alternate the questions between the speakers and may choose to amalgamate questions that cover a similar topic. However, even if we run out of time, all questions will be posted to the speaker on the Q&A and that will be sent to them at the end of the event. The upcoming MycoTalks are listed on our website and you must register for each session. Please note that the time of the talks is 1600 hours UK time. So in the spring and autumn, please check how this might be affected by your local daylight saving status. We would also like to draw your attention to the Medical Mycology Trainee Seminar Series, which is held on a monthly basis, and it's a great opportunity for early career scientists to give a talk. More recently, we've launched MycoClinics as a series of online talks, specifically with a clinical focus. Again, you can find the schedule for these on the MRC Centre website. So today's speaker, the first speaker is David Denning, and he's an infectious disease clinician with expertise in fungal diseases. As Professor of Infectious Diseases and Global Health at the University of Manchester, he managed the National Aspergillosis Centre from 2009 to 2020. Through his lead role in LIFE, which stands for Leading Internal Fungal Education, and as the Chief Executive of the Global Action Fund for Fungal Infections, known as GAFI, he has played a crucial role in improving patient outcomes through online education and advocating for universal access to fungal diagnostics and antifungal therapies. So welcome, David. We're really looking forward to your talk.
Thank you very much for the introduction. So I'm going to talk today about how we can minimize deaths from fungal infections around the world. I'm going to do this almost exclusively from GAFI perspective, that is to say an advocacy perspective informed by science, mostly epidemiology and clinical science. We set GAFI up in 2013. It's really trying to act as a voice, particularly with WHO and other global health agencies, about the need to improve access to diagnosis and treatment across the world for multiple fungal infections. And it's gratifying to see that there's been a lot of progress, broadly speaking, at the WHO, but also amongst other global health agencies in considering fungal disease as is something that's worth investing in. One thing that was a surprise to me a little bit was that the University of Manchester applied for the a sort of university scoring system or a league table related to impact on the Sustainable Development Goals, which the WHO and UN produced some years ago. And number 3 of those goals is related to health, improvement of health. And our submission on fungal disease, and particularly aspergillosis, was the one that was used in last year's application. And the University of Manchester came top of the league worldwide. And that tells you something, I think, about the importance that is perceived of this topic area in terms of improving health worldwide. We think that there are about 13 million people at risk every day of suffering from a life-threatening fungal infection. It's a difficult number to be sure about because there's lots of different host groups at risk and different stages of disease. So leukemic patients may be at risk only when they're neutropenic and not otherwise, for example, and HIV patients only when they have late-stage disease. So the number is somewhat uncertain, but it's a pretty large number. And we think there are about 2 million deaths worldwide each year, which are related to fungal disease, but they're hidden under other causes of death. And you can see this as the ranking of causes of death from 2016, and cardiovascular deaths, ischemic heart disease and stroke top the table, and COPD or smoking-related lung disease or related to mining is number 3. And then you've got various other causes of death coming down here. And the arrow, the green arrow, represents where fungal disease deaths may occur and are a contributing factor to those deaths in that. And this gray bar in the middle shows you, we think, where fungal disease would fit if it was counted as the primary reason for death in many of these patients. So we've also done some modeling about how many of these patients there are in different categories. So if you take tuberculosis and how many have chronic pulmonary aspergillosis, and how many deaths there are and how many lives could be saved, we've categorized these here. In fact, I think based on newer evidence that we have, this is an underestimate for mostly aspergillosis complicating TB. In leukemia and lung cancer, you've got a similar number. There are about 300,000 to 400,000 asthma deaths every year. These tend to be in patients with fungal asthma. And so that group of patients is probably salvageable in part. You've got a lot of patients in ICU across the world with candidemia and invasive aspergillosis. And then in the recent COVID outbreak, these numbers went up a lot, but nonetheless, quite a lot of these patients are salvageable. In HIV/AIDS, the number of patients who die of a fungal infection is nearly half those who die overall with HIV/AIDS, and that's mostly a salvageable number. And then there's an awful lot of patients with chronic pulmonary disease, COPD, who get invasive aspergillosis or are sensitized and infected with aspergillus with CPA. And that number of patients that could be salvaged is also quite large. So overall, this gives you a sense of the scale here. And there are probably about 1.3 million lives of the 2 million lives that could be saved with our current diagnosis and therapy. We don't have perfect diagnostics and we don't have perfect therapies, but this, with application of what we do have, this is what we could achieve. So in terms of thinking about how one positions this for those who know nothing about fungal disease, and it has to compete, if you like, with other topic areas such as malaria or COVID or hepatitis or breast cancer, how does it pan out? Well, you've got around 200 deaths per hour due to fungal disease. Most fungi are not transmissible. There are exceptions to that, and the dermatophytes and pneumocystis are exceptions, but the majority are not. And they usually, most of these diseases happen as complications of other disease, particularly lung disease with TB, asthma, COPD, which is an airborne infection primarily, those with immune deficits, which may be semi-permanent, as with late-stage HIV, or temporary, as in neutropenia or patients in ICU with immune paresis. And these can be either airborne or gut, or sometimes hospital-acquired infections. And then you've got patients who, without immunodeficiency or other defects, who get an injury in the skin or eye and ending up with mycetoma or fungal keratitis. The patient populations are pretty fragmented in many small categories. Many of them are underprivileged, and they don't really have a voice because of that. And patient engagement, because some of these infections are very lethal, and the patients are really not able to voice their concerns about it. And really, it's the chronic Aspergillus patients that have some voice, but otherwise we have very limited patient engagement. The new rapid diagnostics are important and really do transform the ability to make a diagnosis, but the access across the world is poor, with perhaps 25% across Africa and low and middle income countries. But we do know that early detection of these infections is vital for minimizing morbidity and survival. And we also know that culture and microscopy are not good enough. There are many, many false negatives with these older systems. We also have a problem with access to antifungal medicines, as you well know. So amphotericin was first used the first published case was in 1959 for a patient with chronic pulmonary aspergillosis in Michigan. And we've had flucytosine since the mid-70s, but still the access is pretty poor. And then there's a generic problem, and those of you on the call can definitively contribute to this, is a lack of skills and knowledge. Many of these are lack of clinical skills, but there's also a lack of radiology appreciation of the disease. So patients with fungal lung disease are usually not called out, and tuberculosis is the most common differential diagnosis, and that's what's mentioned on the result and not a fungal disease. And there's also a lack of skill in the laboratory. Sometimes the skill levels don't need to be high, as in the cryptococcal antigen testing system and others, but for identifying unusual fungi, there's a very, very high level of skill required. So I now want to tell you a little bit about our program in Guatemala, which we've been running since 2017. It took about a year and some to set up, and it's with a foundation there, the ASI, which have been remarkably effective at implementing. And what we're going to show you is that this can really quite quickly improve patient outcomes by reinforcing, automating, and centralizing access to diagnosis combined with clinical training. So we have the old landscape of diagnosis of fungal disease with microscopy, culture, and pathology. These days we have simple antigen and antibody tests, which may be lateral flow or ELISAs. We have PCRs for many different pathogens, including and commercialized for pneumocystis and aspergillus, for candidemia, trichophyton, and coming up are tests for mucorales. And we have, of course, rapid identification systems with Mordatov. But the antigen antibody testing systems do improve sensitivity considerably, and they certainly improve speed as well. And as a general statement, for life-threatening infections, the patient can afford a false positive test, but not a false negative one. And so what we're looking for are tests that, if you like, pick up all the patients, even if some patients are overcalled, or it turns out to be a false positive. So in the Guatemala program, we brought into the group, into the study, new patients with HIV, those who had abandoned antiretroviral therapy for at least 90 days and were looking for care, and those who were on antiretroviral therapy also looking for care because they didn't feel well. And this program was run in 13 centers, so there was a lot of coordination required to make this happen. And importantly, these patients are screened. So this wasn't simply a matter of the doctor thinking, oh, I wonder if it's this infection, or I wonder if it's that infection. They got blood and urine and sputum obtained regardless of their CD4 cell count, which gives an indication of their immunity. And they were, once the screening tests were done, they were then followed up for 6 months. And the focus was TB, histoplasmosis, and cryptococcus. And later we've added pneumocystis, but there wasn't enough funding to support pneumocystis PCR early in the program. So as the program was getting going, an analysis of histoplasmosis in the Americas was conducted by Anita Sil in French Guiana, and he looked at all of the literature was there and postulated that the rate of histo in late-stage HIV was 4%, 4.16%. And what the Guatemala program did was to use histoplasma antigen testing. And this is a recent meta-analysis which really shows that it's a very effective test with a 95% sensitivity and 97% specificity done on urine. Now, some patients don't produce urine, so that's a limitation. It can be done on serum, but there are very few diagnostic tests with this level of performance. So we've written several papers now about this program, which we've called the Diagnostic Laboratory Hub as the core of this. And these are the centers, and this is the drive time or courier time from each center to deliver samples to Guatemala City. And there was online ordering of tests, so the lab knew when the tests were coming, a courier service, there was a mobile phone alert for results, and then an online result portal so that the local doctors could see the results immediately, and it didn't require, as required fairly recently in the UK, a postal service. And what this shows, our program showed, if you look at this, is that the number of patients screened was about just over 2,000. That's the green bars here per year, 2017. And as time went on, more and more opportunistic infection cases were diagnosed as doctors felt more comfortable with the program and sent more samples, because the more samples you get, the more likely you are to make a diagnosis. And the mortality fell from about 25% down to about 16.5%. So that's an 8% fall. And then we had COVID that arrived, and the patients didn't attend the hospital. Fewer samples were sent. It was more difficult to get them to the lab, and the mortality went straight back up to where it had been before, telling you that this is effectively a natural experiment of that the importance of continuous rapid diagnosis is really a very important part of the program. And when you look at the reasons for death, you can see that there's a slight increase in numbers of samples over this time frame. The deaths have gone down by 8%, as I've indicated. And most of the reason for the improvement in survival was actually TB deaths that went down. And what was happening before was that patients were thought to have tuberculosis. They were being treated for tuberculosis and then they died. Whereas if you actually make an accurate diagnosis, you don't call them TB, they're treated for the right infection, and actually the outcomes for the patients who actually do have TB are better than was perceived to be the case before. So overall, of the new patients coming through who'd not been on antiretroviral therapy before, 18% had a life-threatening opportunistic infection, not including pneumocystis, which is usually around 15% as well. And when you look at the distribution of those infections, this is done by CD4 cell count, which gives you an indication of the immune status of the patient. You can see that actually the largest group of patients were patients with histoplasmosis, and the rate was 7.9%, so almost double the anticipated rate that Antoine had published. And that tells you that if you go looking for these infections, you find them, and that also, particularly in the low CD4 cell count group, you find them. But you also find infection of all of these infections actually at above 200 CD4 cells, because this program actually had patients in with 200 and 300 and more CD4 cells. And so that the 200 cutoff is not absolute, and we need to be wary of that in terms of thinking about looking after the patients. And you also had a group of 8% of patients with dual or triple infections, and these were particularly difficult to treat and had a worse outcome. And if you hadn't been screening for these infections, you probably would have missed these dual or triple infections because you'd only be focusing on one. If you look specifically at histoplasmosis, you've got the 3 years here, the number of patients screened up here for histo. So this is one of the groups of patients where you had more histo testing as over time. And you've got quite a lot of new patients appearing with the different, with new patients with HIV coming through. And you can see that rise in a diagnostic yield. And then also this diagnostic yield in those who return to care having dropped out of care. And when you look at the survival of histoplasmosis, just as you've seen with TB, or we've seen with TB, you can see that you've got a fall of about 11%, 11.5% in newly diagnosed HIV patients with not dying of histoplasmosis, a similar fall of those on ART, but actually a slight sort of uncertain and slight rise in those who are returning to care with it. And we don't really understand this shifting of this baseline and what that means. But what is gratifying is that actually you can save really quite a lot of these lives. And this is in the context without liposomal amphotericin, which is one of the things that's necessary. So what this style of health system offers is it offers commercial techniques in the laboratory. So they tried to implement non-commercial techniques, but it was really difficult to get those to work properly. There was a lot of variability. And we really do need to focus on tests which are commercially available and well validated. You've got quick implementation because they had a, what you do is you take existing laboratory facilities and you add tests to them. It's pretty cost effective in terms of human resources and automation. You've also got a continuous means of educating healthcare workers, partly through seminars and online working, etc., but also because they're getting results all the time. And so they're learning as they're going along about the patterns of disease that patients have got. You've got a fairly straightforward means of doing quality assurance and control and proficiency testing. You've got definite value to the country in terms of registration of tests and drugs. And you've got increased contributions and awareness by pharma and diagnostic companies who are more willing to invest in education and other elements that are useful for patients and doctors. And of course, as I've shown you, you can collect data and act as a surveillance hub. So we think that this model of diagnostic approach would be very helpful in many other settings. Now, it may not be obvious to you, but actually histoplasmosis is a lethal disease with late-stage HIV unless it's treated. This is not a survivable problem. And so it's really very important to diagnose these patients. As I've indicated, the antigen is really very good. Intravenous amphotericin is the usual thing. And this study done by Johnson et al. In the US published nearly 20 years ago shows that liposomal amphotericin is a bit better than conventional amphotericin B, with a p-value here with a 2-to-1 randomization ratio. And this work has never really been repeated and needs to be, and we need to roll liposomal amphotericin B out to many, many more centers than we have at the moment. The only other indication where liposomal amphotericin B is superior to conventional amphotericin is in visceral leishmaniasis. Otherwise, it's less toxic, but a higher dose has to be used. And once you've got better from histoplasmosis, you're better. You know, you can go on and lead a normal life, which is slightly different from cryptococcal meningitis, where there are sequelae related to the disease, unfortunately. And once the patients have been started on ARVs, then and the CD4 count rise to 200, you can then stop therapy. The alternative in medication is itraconazole. Of course, it interferes with rifampicin or it is interfered with by rifampicin, and that's a challenge in some patients, and also with some ARVs. So as part of our advocacy work and working with CDC and PAHO and WHO, we contributed to meetings around enabling guidelines on histoplasmosis to be issued. And these are those guidelines issued in 2020. And I won't go into the detail of these about how you treat the patients, but the idea here is that every country which has got endemic histoplasmosis should have antigen testing and should have treatment available with amphotericin and preferably liposomal amphotericin. Which brings me to Africa. And Rita Oladele, who is back in Lagos, having completed her PhD with us in Manchester some time back, has just coming to the end of a study which she presented at the TIM meeting of antigen testing in Nigeria. And you can see the map here with the different rates. And the different rates vary from more than 10% around Ibadan down to about 1 to 3% of late-stage HIV patients with disseminated histoplasmosis in the context of AIDS. And this is really completely new. We have really had very, very little information on this topic from Africa. There have been a couple of surveys from Kampala and one from northern Tanzania with quite low rates. But these rates of more than 10% are pretty high. And this is using the IMI-EIA assay. And that compares with rates of cryptococcal meningitis. And if you look at cryptococcal meningitis, this is the mean here for all these different countries, and I won't go through all of it. It's about 4.5%. So that's the rate of crypto, mostly in Africa. Here you've got histoplasmosis rates, which are double that. And yet we don't look for this. It is just not part of the current approach to looking after patients with HIV in Africa. And we're in the process of coming to the end of a survey of diagnostics in Africa, which Emma Orafua and Richard Penny have been doing on behalf of Gafi. And in this map, orange means not available at all, and light blue means barely available. And so you can see that there's almost no availability of testing at the moment in the whole continent. The only country that says it's got it is Zimbabwe, but we think there's an issue there around fear of being negative about your country, given the rather autocratic regime that's in place in it. So this is a really something that needs really quite radical thinking to take forward. And Africa is not alone in this problem. We published a couple of years ago a map of all of the skin testing, or the published cases of histoplasmosis in Africa and skin testing. And this is the map of the published cases. You can see there's quite a lot of cases in Thailand and a few others scattered around different parts of Southeast Asia. But if you compare that with exposure, which is done by skin testing, they're very different. So Myanmar has a hyperendemic area and it's got a lot of histo. We've got northern Philippines with a lot of histo, bits of Indonesia with a lot of histo, and then other areas where there's no data. And this mismatch between where the cases are and where the exposure is tells you that there's a very large diagnostic gap in Southeast Asia. So these countries need, definitely need histo antigen as well. And then I want to finish on a point about tuberculosis. As you will know, about 45% of TB cases are diagnosed clinically based on cough and chest x-ray, but without confirmation in the laboratory. And a question arises as to whether actually these patients actually do have TB or not. So on the left-hand side is a typical patient with TB with shadows there, soft shadows. In the middle, you've got chronic pulmonary aspergillosis, and on the right, you've got subacute invasive aspergillosis with shadows on both sides actually there. And they're pretty similar. And it's not, if you haven't got tests, it's not easy to be sure what the diagnosis is in all of these patients. So Findra Satyangram from Indonesia has completed her PhD with us on looking with a cohort of patients, 200 patients from Indonesia who were looked at. And the question we asked was how many of them have TB, sorry, how many of them finish their TB treatment and have chronic pulmonary aspergillosis. So you can see the challenge here. So the chest x-ray suggests they've got TB in about half. You've got symptoms compatible with aspergillosis in a third, and you've got an Aspergillus antibody test that's positive in about 40%. But in order to diagnose CPA, you need all three of these elements. And when you do that together, you end up with about 13%. But that's a lot of people. At the moment, this isn't even considered as a differential diagnosis at the end of TB. Many, many centers in the world don't x-ray patients at the end of their TB therapy. They just send them on their way. And so this is really very different from where we have been before. There's a new lateral flow assay out, and this shows it. This is for Aspergillus antibody. On the left, you can see a patient that's positive, another one weakly positive. This is a negative. Then you've got a positive, a strong positive, and another negative. And the performance of this assay is pretty good. When we compared it with the standard UK test that we use, which is Immunicap, it actually was more sensitive at about 92%, and very specific with blood donors that were UK blood donors. When you apply this in a more complex setting with a lot of other lung disease without CT scanning to confirm the diagnosis and some undiagnosed TB patients, you end up with a sensitivity and specificity of about 80 and 70%, which is still pretty good, but not quite as good as when you've got more diagnostics available. And it's only €4 or $4 each, this test. So this is a lot less than a single gene expert test, for example, which is more like $14. And again, as part of our survey in Africa, we have looked at where patients can have an Aspergillus antibody test done. And the answer is many, many countries don't have any Aspergillus antibody testing, and only a few have this regularly. So Morocco, again, Zimbabwe, which we're not quite sure about, and South Africa. And if you applied this testing to everybody who had clinically diagnosed TB, which was not proven in the lab, patients coming back with a relapse of TB, which may not be a relapse of TB, but actually CPA, it would cost about $39 million a year globally using the lateral flow test. So just to contrast that, the amount of money spent just in Kenya on HIV each year is $500 million. So this is not actually big money in the global health expenditure terms. And we've done some work on this in more detail with several studies over the last 7 or so years. So the people who can get CPA includes those who can mimic TB. So these are the HIV negative patients and smear negative patients, and they get it. You get co-infection, as I've indicated, at the end of therapy. And if you estimate that about 10%, although FINDRA's data suggests 13%. And you can get CPA after TB in those who've got cavities. And Ian Page's work in northern Uganda showed that this was 6.5% per year if you have a residual cavity. And then if you assume that you lose 20% of these patients in year 1 of their CPA, which seems to be the current figure particularly untreated, and then a 10% annual death rate, then if you look at the India numbers, they're huge. So India has the largest population of TB in the world, about 2 million cases a year, pulmonary TB, and roughly 500,000 deaths. And if you look at the number who may have chronic pulmonary aspergillosis, you've got roughly about a bit short of 500,000 in these patients, most of them HIV negative. It's not a big problem or as big a problem in HIV positive cases. And a lot of deaths, probably about 180,000 deaths a year, which is a big part of the 500,000 deaths from TB. Unfortunately, of course, all the data that we've used to make this estimation in India is from other countries. And so there is a need, I think, to generate data in each country to be confident of what the answers are. And we don't have really good data on mortality in younger people with CPA, because it tends to be an older population. And so we need that information as well. So where do we go now? Fungal diagnostics and antifungal agents need to be on the WHO essential lists, or they are on the essential medicine list of WHO. Now they need to be available in every country. The epidemiology of these infections is often sketchy. We need much more data. And this is a really good way of engaging clinicians and diagnostic labs in mycology and in writing abstracts and generating useful information to power up this area in their country. We need a major emphasis on implementation research, which tests done where, clinical awareness, screening versus diagnosis, turnaround time improvements, etc. So this implementation research is something that traditionally hasn't been much done in our topic area and needs to be. We also need health economics, and this is an area which has barely been touched for fungal diseases. Our view is to work through existing mechanisms and structures to add mycology to current programs. We don't think it's sensible to build complete new laboratories, complete new services, but we do need well-trained people who are passionate about the topic, because that's how change is brought about. And our audience is primarily currently mycologists, but we need to engage people leading other programs, HIV programs, dermatology programs, chest disease programs, and others. And we think that public engagement is best approached through chronic fungal disease, because it's quite difficult when patients have life-threatening infections to engage them in this way. We've drawn support from multiple different sources over the last 7 years, and we're very grateful to all those who have contributed to our mission. Most of this resource has been to run our meetings. The GILAG Foundation has funded our program in Guatemala. So with that, I will finish. We have a basic problem of too many people without access to life-saving diagnostics and antifungal medicines, and this really does need to change. And what we're looking for ourselves and with others is sustainability, so that long-term care can be provided in all these countries around the world. Many thanks for your attention.
Thank you very much, David. That was a very important talk. And I now am going to introduce Professor Anita Sil, who's going to go on and give us much more information on the cell biology of histoplasma. Professor Anita Sil was an undergraduate at Harvard College and then received a PhD and MD from the University of California in San Francisco. During her PhD, she worked with yeast geneticist Ira Herskowitz, where she discovered ASH1, an asymmetrically localized transcription factor that regulates mating type switching. After receiving her MD, Dr. Sil began working on the thermally dimorphic fungal pathogen Histoplasma capsulatum, which you'll hear about today, first as an independent UCSF Sandler Fellow, and then as a tenure-track faculty member in the Department of Microbiology and Immunology at UCSF, where she's now a full professor. Anita's studies explore the links between temperature sensing and virulence and the molecular communication mechanisms at the host pathogen interface between Histoplasma capsulatum and the macrophage. And today she's going to talk about the regulation of cell shape and virulence in histoplasma. Thank you. Welcome, Anita.
Thank you, Judy. Thank you for that kind introduction. I'll just start my screen share. And I'm very grateful to the organizers for inviting me to speak, and to David for giving such a wonderful entree into thinking about histoplasma biology, as we've heard about the consequences of histoplasma infection around the world. So as Judy said, I'm interested in the basic biology of histoplasma, as well as some of the other thermally dimorphic fungal pathogens that are evolutionarily related to histoplasma. And I love these images because they illustrate one of the main properties of these organisms, which is that in the environment, they grow in a filamentous form. So these are the fungi I'm going to talk about today are all soil organisms that grow as hyphae in the soil. And when the soil is disturbed, fragments of these hyphae, as well as vegetative spores, can get inhaled by mammals, including humans. And in response to the body temperature of the mammalian host, these organisms transition to a host form, which is a budding yeast form in the case of Histoplasma. And in my laboratory, we're very interested in understanding how these cells sense temperature and then turn on a virulence program that's critical for pathogenesis in the host. And additionally, or as part of that investigation, we studied the interaction between these fungi and innate immune cells, mostly macrophages. So I'm going to focus today on this, mainly on this question of how cells sense temperature. And as I mentioned, Histoplasma is one of a group of thermally dimorphic fungal pathogens that grow in a filamentous form in the soil and transition to a host form in response to introduction into the host in the lungs. And this is a switch that we can mimic in the laboratory simply by switching the temperature from room temperature to 37 degrees. I just want to point out, since I'm sure many of you are more used to thinking about Candida or Aspergillus, you know, the difference for these fungi is that really, you know, the filamentous form is largely, although not completely, confined to the environment. And in the host, one finds predominantly a single morphology with some exceptions. And that's in contrast to Aspergillus, which has no yeast form, of course, and to Candida, which is multimorphic in the host. So for none of these organisms here, you know, do we really understand the sensors and the signal transduction pathways that govern, you know, whether how the organisms sense and respond to temperature. And what I'll do is start out talking about Histoplasma, and then at the end of the talk, I'll tell you a little bit about coccidioides. Okay, so quite a number of years ago, a former postdoc in the lab, Diane Inglis, figured out a way to isolate Histoplasma conidia. And she took those conidia and she germinated them either at mammalian body temperature, 37 degrees, or at lower temperatures. And she found that the spores seemed to be intrinsically temperature responsive in the sense that the spores germinated at 37 degrees gave rise, the majority of them gave rise to yeast cells, and the spores germinating at lower temperatures, the majority gave rise to hyphal or filamentous cells. And this was I like these images because they highlight that, you know, these are the same populations of spores, but they behave very differently in response to temperature. Now, once those spores germinate to give rise to yeast, either, well, let's say in the context of an infection, those spores and the resultant yeast cells are likely to encounter cells of the innate immune system, including macrophages. And we've done a lot of work looking at the interaction between Histoplasma, both spores, but mostly yeast cells, and a model macrophage, murine bone marrow derived macrophages. And I want to point out to you here that that transition to the host form is accompanied by the ability of the organism, of the fungus, to now colonize and lyse host macrophages, which I'm going to show you that host cell lysis in this movie. So these are bone marrow derived macrophages that have been infected with Histoplasma yeast cells that are producing a secreted M-cherry protein. And each Histoplasma cell is enclosed in its own phagosomal membrane. And so that secreted M-cherry protein is ringing the phagosome. It's accumulating at the phagosome membrane, and that's why you see all these oval shapes inside the macrophage. And as you can see, what happens at a particular point during this conversation between the intracellular fungus and the host cell is that the macrophage undergoes lysis. And in my lab, we study those fungal factors that are important for this communication and this lysis of the host cell. Okay, so I'm going to focus for the first part of this talk on this question of how cells sense temperature and turn on yeast phase growth and virulence factor expression. And we've been studying this for a while, and we've found, that temperature, as I mentioned, is a key signal that governs this transition between the soil forms where you have hyphal cells and vegetative conidia and the yeast form of the organism. And when you examine these states, the filamentous form and the yeast form, you see that there are gene expression changes. There are robust changes in gene expression. Say, 20% of the genome or more is differentially expressed between the two forms. We published a few years ago that some of the transcripts undergo alternative transcript start site selection when you compare the filamentous form and the yeast form, which is a very interesting form of regulation. And we have some evidence for translational regulation as well. So ultimately, temperature is triggering a number of molecular events that result in a cell shape change and the expression of factors that promote host colonization and disease. So we imagine, in a simple-minded hypothesis, in terms of how the cells sense temperature, that there must be some molecule, or more likely, I think, set of molecules that change their conformation in response to temperature and act as molecular thermometers that then govern the remainder of the signal transduction events at each temperature. And so we imagine that the conformation of these molecules determines whether the cells go down the pathway of filamentous growth or yeast phase growth. And perhaps even these pathways, as we elucidate them, would be mutually antagonistic, so that once the cell makes a decision to go down one pathway, it's essentially turning on mechanisms to block the other pathway, and vice versa. So a number of years ago, we did a genetic screen for genes that are required for yeast phase growth. And so we reasoned that it would be possible to identify mutants that couldn't grow in the yeast form and instead were filament locked. And we indeed found such mutants. So these are images generated by Sinem Behan, a very talented former postdoc in the lab who now has her own lab at JCVI. And what we're showing here is that in contrast to wild type cells, which grow in the yeast form at 37 degrees and make filaments at room temperature, each of these mutants, which we call RIP mutants, are constitutively filamentous, independent of temperature. And each of these mutants denotes a different gene, RIP1, 2, 3, or 4. And these genes are named RIP because they are required for yeast phase growth. Now, each of these RIP genes encodes a transcription factor, and these are conserved fungal transcription factors. So RIP1 is conserved throughout the fungal kingdom, and it's an ortholog of WAR1 in Candida, the regulator of white opaque switching. So in many fungi, the RIP1 orthologs have been shown to be key developmental regulators. RIP2 and RIP3 are members of the same family. They are velvet proteins. Their orthologs in Aspergillus are named here. And again, in Aspergillus, these are important developmental regulators that govern cell fate in response to light conditions instead of temperature. And then RIP4 is the ortholog of FACB, which governs acetate utilization in Neurospora and Aspergillus. So in histoplasma, we have found that these transcription factors have been rewired to govern the ability to grow as yeast in response to temperature. And I'm not going to show you all the data today in the interest of time, but we have seen that all 4 of these RIP factors are upregulated either at the transcript level or the protein level, or both, at 37 degrees. And I will show you on the next slide a heat map that illustrates that each of these transcription factors is required for the normal transcriptional program at 37 degrees. So you can see that here. This heat map is comparing in the first column wild type cells grown in the yeast form at 37 degrees to filaments at room temperature. And you can see, or CNM found that there's a set of genes that are enriched in their expression at 37 degrees, and a set of genes instead whose expression is correlated with the filamentous form. And then if you compare the transcriptional program of each of the RIP mutants at 37 degrees to wild type, you can see that these mutants fail to turn on the normal complement of genes that's expressed at 37 degrees. And instead, they inappropriately express genes that are normally only expressed by, or that show enriched expression in the hyphae. CNM went on to look at the direct targets of each of the RIP transcription factors. And I'm showing you here a site escape diagram of the targets for RIP1, RIP2, and RIP3. Yellow on this diagram indicates enhanced expression at 37 degrees in the yeast form, and blue indicates this filament associated expression normally in wild type. And if you look at the direct targets, there's a couple of take-home messages. So each of these transcription factors has some unique targets, but there are shared targets that are enriched for genes that show temperature regulated expression, either the yellow or the blue. And this set of genes also seems to be enriched for the small number of virulence factors that we know about. So this has been a really great way to mine the genome for new potential virulence factors. And that's ongoing in the lab. And I'll come back to that closer to the end of the talk. Okay, so one of the sort of great findings for us was that this work told us that in response to temperature, we now had these 4 molecular handles, these transcription factors that are upregulated at 37 degrees. And these transcription factors, if you look at their direct targets, seem to regulate genes that are required both for the cell shape change at temperature and the expression of virulence genes. And so they're the first molecular link between temperature and these phenotypes. So again, I'm not going to show you the work today, but we know that at least RIP1, 2, and 3 form a biochemical complex, and they're part of a circuit that is clearly required for the normal cell shape and molecular responses to temperature. But we'd really like to understand the regulation of this pathway, and we're exploring that in a number of ways. But I thought I'd highlight next some of the work from my lab on how this program is blocked at room temperature. So we thought that one way of understanding its regulation would be to see what keeps it off at room temperature, and that might shed some light on what's required to turn it on at 37 degrees. And so a former postdoc in the lab, another very talented scientist, Sarah Gilmore, started a screen and did a pilot screen for genes that are required for filamentation in response to low temperature. So in this scenario, we're looking for mutants that can't make filaments at low temperature. In other words, mutants that are locked in the yeast form independent of temperature. And Sarah found such a mutant, and this mutant was further characterized by Lauren Rodriguez, a very talented former graduate student in the lab. And they found that one of the mutants from Sarah's pilot screen, one of the yeast lock mutants, has an insertion in a gene that we're calling MSB2. And I'll tell you in a moment on the next slide what MSB2 is, but I want to show you the striking phenotype, which is that if you take wild type yeast cells at 37 degrees, and I've already told you that they form filaments at room temperature, And in fact, when you shift these cells to room temperature, the yeast cells really seem like they don't divide well, and they stop, and over a period of days make this transition to room temperature. This MSB2 mutant is yeast locked, and in fact, when you shift the yeast cells to room temperature, they don't seem to notice, and they continue to grow happily in the yeast form. So what is MSB2? So MSB2 encodes a signaling mucin. So signaling mucins are, these are extracellular proteins that have a mucin-like extracellular domain that's highly O-glycosylated. And they have a cytosolic signaling domain that can interact with other signaling proteins, for example, MAP kinases. So our MSB2 in histoplasma is named after its more famous orthologs in Saccharomyces cerevisiae and Candida albicans, where it's involved in the response to high osmolarity as well as filamentous growth pathways. And so we were very excited by the fact that a protein whose orthologs are implicated in filamentous growth was a protein that we found to be required for filamentation in histoplasma. So because we're interested in regulatory circuits in response to temperature, Lauren wanted to see if MSB2 is required for the normal transcriptional response to low temperature. And so she took wild type cells, the MSB2 mutant and the complemented strain, all grown at 37 degrees, and she shifted these cells to room temperature. And this transition, as I mentioned, occurs over the course of days. And you can see that in the wild type, in the case of the wild type cells and the complemented strain, that you get filamentation in response to this temperature shift. But as I've already told you, the MSB2 mutant is yeast locked. And so Lauren did RNA-seq at all of these time points. And what she found, to our initial surprise, was that in the MSB2 mutant transcriptionally looks actually very much like wild type and the complemented strain, even though it's not undergoing filamentation. So there are several thousand genes that change in expression, either yeast-specific genes that get turned off when you shift to room temperature, as well as genes that turn on when you shift to room temperature. And the bulk of those change in expression, just like wild type in the MSB2 mutant, except for a very small regulon of about 165 genes that are normally turned on or induced when the cells filament at room temperature, and a small regulon of about 40 genes that normally gets turned off when the cells switch to room temperature and start filamenting. So this work is published, and I don't have time to tell you about all of it in detail, but I did want to emphasize that this work, the fact that there are only a small number of genes that show MSB2 dependent expression was very useful for us, because in the case of these genes, and these are multiple time courses when cells are shifted from 37 degrees to room temperature, these genes that turn on in wild type and the complemented strain, but fail to turn on in the MSB2 mutant, are implicated in filamentation because this is a mutant that fails to filament. And in fact, Lauren went on to validate that one of the genes in this regulon, HOG2, which encodes a MAP kinase, is indeed required for filamentation. So if you knock down HOG2 by RNAi, or as shown by Dror Asa in our lab, if you mutate it using CRISPR, those mutants are yeast locked. So now we have just instead of thinking about the thousands of genes that change in expression, we can focus our attention on a smaller number that are associated with filamentation. And then the other regulon of genes that normally turn off when cells shift from yeast to filaments at room temperature is similarly interesting. So there's a set of genes that turns off in wild type and in the complemented strain when you shift to room temperature, but that those genes fail to turn off in the MSB2 mutant. And so these are now transcripts that seem to be associated with growth in the yeast form, independent of temperature. And they are enriched for secreted virulence factors, like the virulence factor CBP1, which Bill Goldman's lab and my lab has studied for its role in pathogenesis, as well as transcripts that are direct targets of those RIP transcription factors. So this, again, narrows us down to 40 genes that whose expression is associated with growth in the yeast form, including known virulence factors. And this has been an important data set to mine to find novel virulence factors again. And I'll just give you an example of that, there are 2 transcripts here that are indicated with an asterisk, and those transcripts are part of a family of proteins called knotins. So knotins are cysteine knot proteins. There's a knot in the protein that's formed when there are 3 disulfide bonds in the knotins, and one of those bonds passes through a loop formed by the other 2 disulfides, and that generates a very stable knotin fold. And these proteins are found in animals, plants, and fungi. They're common in invertebrate toxins. And in histoplasma, the knotins have undergone a large expansion. So these are secreted proteins in histoplasma, or at least predicted to be secreted. While many fungi have 0, 1, or 2 knotins, as I said, they've undergone an expansion, and 2 of them are regulated by MSB2 in the sense that their expression is correlated with growth in the yeast form. So even at low temperature in the MSB2 mutant, when the cells are growing as yeast, these knotins are still expressed. And so Dinara Azimova, a very talented graduate student in the lab, mutated one of these knottin proteins. And here what you're seeing is a macrophage monolayer lysis assay where if you take macrophage monolayers that are uninfected over time, the density of the macrophages increases and you can stain with methylene blue and see a confluent monolayer. In contrast, if you infect with wild type histoplasma, the histoplasma will clear the monolayer and you can see that this knottin mutant is defective in monolayer clearing. And Dinara has gone on to quantify this defect, and we can see that this gene is required for optimal macrophage lysis, and we're interrogating the potential function of these very interesting knotins. So I did motivate this work by telling you that we wanted to understand how the RIP pathway was regulated and what keeps it off at room temperature. So these are Western blots looking at RIP1, RIP2, RIP3 protein and a loading control in wild type cells grown either at room temperature or 37 degrees. And you can see the accumulation of these proteins at 37 degrees. There's very little at room temperature. In contrast, in the MSB2 mutant, RIP levels are inappropriately high at room temperature. And that tells us that MSB2 is required for keeping the RIP protein levels at least low at room temperature. In the opposite direction, we think that at 37 degrees, the RIP factors inhibit the MSB2 program. So specifically RIP3, this is a chromatin IP track showing that RIP3 binds upstream of the MSB2 gene. And in the absence of RIP3, you get inappropriate expression of MSB2 at 37 degrees. So it's expressed a little bit more highly than it is in wild type cells at 37 degrees. And that's also true of the some of the genes in the MSB2 filamentation regulon, like this transcription factor, STU1. So the RIP proteins are important for inhibiting the MSB2 pathway at 37 degrees, and MSB2 is important for inhibiting the RIP pathway at room temperature. So we think now that morphology is controlled by these 2 antagonistic programs that at room temperature, the MSB2 program dominates and you get filaments. At 37 degrees, the RIP program dominates and you get yeast, and that there's mutual inhibition, as I've just shown you, of these two programs. And we think that somehow in response to temperature, there are molecular sensors that toggle the switch in favor of either one program or the other. Okay, so I want to move to telling you a little bit about coccidioides in the remaining 4 minutes or so. But I'll just tell you that this work that I've told you about in terms of the RIP pathway and the MSB2 pathway has been, really informative about elucidating the regulatory circuits that drive these different developmental stages, and for honing our understanding and our focus on particular genes that might be important for virulence in terms of interaction with the host. Okay, so coccidioides is another one of these thermally dimorphic fungal pathogens. And it's really a little bit more evolutionarily distant from histoplasma than blastomyces and paracoccidioides. But we're very interested in its biology and seeing the contrasts and the comparisons between coccidioides and histoplasma. So this graphic from the Centers for Disease Control in the US shows you the biology of coccidioidomycosis. So again, soil pathogen, the coccidioides filaments, every other cell can disarticulate from the filament and become a viable arthrochonidium, which can get inhaled by the host, and inside the host undergoes a dramatic conversion to a host form called a spheruole, where the spore enlarges, the nucleus of the spore undergoes multiple divisions, this structure then segments, and then can lyse in the tissues to give rise to these so-called endospores that can disseminate. So we'd like to understand which regulons drive temperature-dependent developmental switches in coccidioides, and we'd like to understand the conversation between coccidioides and innate immune cells. So in collaboration with Ali Mandel and in Mark Orbach's lab at the University of Arizona, we've looked at the phenotype of the RIP1 mutant in coccidioides. So the transcription factor that's orthologous to our RIP1 in histoplasma is required for the formation of the host form in coccidioides, the spheruole form. And these RIP1 mutants, Ali and Mark have shown, along with Lisa Schubitz, these RIP1 mutants are avirulent in the mouse model of infection. Interestingly, when Sinem, when she was in the lab, looked at the transcriptional program of the RIP1 mutants in coccidioides, we found that there were genes that were either in wild type cells spheruole or hyphal enriched, and RIP1 was actually required for expression of some spheruole enriched transcripts and some hyphal enriched transcripts. So RIP1 in coccidioides clearly plays a role in both spherulation and in hyphal biology as well. So it's early days for us yet, but we're very interested in elucidating other components of the regulatory network, as well as the targets that are important for this transformation to the host form in coccidioides. And this question, as well as others, are being pursued by some current folks in the lab, Keith Walcott and Christina Homer. And then I'm just going to finish by telling you a little bit about the conversation between coccidioides and our favorite model macrophage, which is, again, bone marrow-derived macrophages. So Jane Symington, who's a pediatric ID fellow in the lab, has been looking at the interaction between arthrochonidia and bone marrow-derived macrophages. And she's developed a nice assay where she labels arthrochonidia with FITC, infects macrophages, and then before fixing the monolayer, stains with chalcofluor white, which then only can access arthrochonidia, which are outside of the macrophage. So green arthrochonidia are internal, and ones that are ringed in blue are still outside the macrophage. And you can see some of the data here. So Some of the spores are green and don't show any calcifluor staining, and others are, like this arthroconidium over here, are both green and blue, indicating that that's outside the macrophage. And you can, of course, quantify these data, and Janie has found that about 50% of bone marrow-derived macrophages after 30 minutes or so contain arthroconidia. Interestingly, if you take arthroconidia and you just put them in this macrophage media, they will make hyphae. But in the context of being with the host cells, we are seeing spherulation. And some of these early spherules that are still small appear to be intracellular, as you can see here. So again, it's early days for us, but we are very interested in seeing, as other people have seen with other types of macrophages, that we see phagocytosis of arthroconidia. And we'd like to understand more about the fate of both the fungus and the host cell in this context. We know that the bone marrow-derived macrophages are promoting spherulation, and we'd like to understand what host signals are required for that and how the fungus is responding. And Jane is also looking at the host transcriptional response to coccidioides infection. So I want to thank by acknowledging the people who did the work. So first of all, I talked about Lauren's work on MSB2, and that mutant was originally identified by Sarah Gilmore. And I talked about Seenam's work on the RIP pathway. I do want to mention Jesse and Basti, who initiated all the coccidioides work in the lab with some help from Lauren as well. And then our current team Cocci, I told you about Jane's work with the arthroconidia and bone marrow-derived macrophages. Keith Walcott has been instrumental in all of our coccidioides culturing and molecular genetics, and Christina is pursuing a number of new avenues in cocci biology in the lab. And I want to thank collaborators in the cocci world, especially Ali and Mark, who did the work with us on the RIP1 gene. Okay, I'll stop there. Thank you very much for your attention.
Thank you very much indeed, Anita. Lovely talk. Okay, so now we'll start our question and answer session. So I'm going to kick off with some questions for David. So Sarah Ali asks David, do you think if we look for fungi alongside standard bacterial culture, especially for respiratory samples, can we raise awareness and increase the diagnosis of fungal infections?
So the answer, of course, is yes in broad terms, but bacterial media are not as effective at growing fungi as fungal media are. The yield for Aspergillus, which is best documented, is about a third lower, for example. And some fungi are special because they require special culture systems and times, and also are category 3 pathogens such as cocci and histo. And histo grows really slowly, so you'd have to keep those cultures going for 3, 4, 5 weeks to keep to grow that. Cocci will grow earlier, but it's significant risk to the laboratory staff. So the answer is yes, but we also need to encourage people to ask for a fungal culture specifically, I think.
Yeah, okay. Yeah, that's a very good point about the length of time. And a question from Ted White. I'll try and shorten it a little bit. There are several parts to it, but basically he's interested in whether there's any data on Talaromyces malnefiae, formerly Penicillium. Is there any impression that the two are causing co-infections? And do the areas of high incidence overlap or exclude each other? Right,
so the areas of incidence do partially overlap in Southeast Asia, but of course histo is much more widespread than that. First thing. Secondly, I haven't seen much data on co-infection. Their clinical presentation is fairly similar. You can get skin lesions in the context of late stage AIDS with some respiratory disease and lymph nodes and so on. And so I think that you probably get one or the other. Whether patients would be at risk for both, whether the same patients, I don't know. I don't think we know the answer to that. But in the Southeast Asia, both of these infections need to be looked for actively. And they both produce galactomannan, actually, so you can diagnose them with Aspergillus antigen testing because of the galactomannan cross-reactivity in some patients, although it's not as sensitive as having a specific test for them.
Okay, right, thank you, David. Over to Judy.
Okay, so we have a question from Mukund Ramakrishnan for Anita. And he said, it's really interesting that one of the transcription factors regulating the yeast to hyphal transformation is VEL-B. Does exposure to light of any kind play any role in the yeast to hyphal switching in histoplasma or in yeast?
So light exposure does seem to play a role in the switch, and there's some old literature that suggests that dark conditions favor yeast phase growth, and this is being studied extensively by Sinem Behan in her lab at JCVI, and Sinem has done some very interesting work looking at sort of the kinetics of the switch in light versus dark and the gene expression program, and has clearly validated that it does seem to indeed be the case that darkness can facilitate yeast phase growth. But what the players are in that process, we don't actually know. So,
okay. There's also a question from Ted White. He says, very nice talk. Are the RIP genes in histo additive, or is the phenotype maxed out with just one of them? And in cocci, does infection of the RIP1 mutant protect mice from later infection with the wild type?
So the RIPs are not additive, and that is most likely because, as we know, each of the RIPs regulates the others. So if you're missing any one of them, you're really missing all four of them. So there's positive feedback loops where, so let's see, RIP1, RIP2, RIP3, and RIP4 bind upstream of RIP1, RIP2, and RIP4, and there's all kinds of interregulation, but the main point is you pretty much lose all of them when you mutate any one. And in coccidioides, interestingly, so the RIP1 mutant is not protective. So unlike some other cocci mutants where you can infect the mice with those avirulent mutants and then see protection from infection with a virulent strain, that isn't the case with RIP1. And maybe it's not so surprising because the mutant doesn't undergo the key transition to the host form. And so maybe it fails to produce the, spherical type or antigens that would be important for that protection.
Okay, Alex?
Okay, so a question for David from Honster Koch. Can you expand a bit, please, on what you mean by health economics?
Right, so if you're let's imagine you're in the Ministry of Health and you've got a pretty limited budget. You might be able to get money from the Global Fund or PEPFAR or some other agency to pay for stuff for 2 years or 3 years or some period of time, but you might not be able to. And you're faced with mothers having catastrophic pregnancies, babies dying, or diagnosing aspergillosis or diagnosing histoplasmosis. What do you put your money into? How do you make a decision? So we do know that the people who die of cryptococcal meningitis and histoplasmosis in AIDS are about 35 years old, right? So I don't know if you were designing society. For me, losing 35-year-olds is a complete disaster for your society because these are usually parents, they're in the prime of life, running economically, building businesses or schools or whatever they're doing. So this is really, really a bad age to lose people. So you can do fairly simple health economics and say, well, if I tested this many people, this is how much it would cost me to test it. And you can compare that, for example, with giving vaccines to children or putting in new wells for water or something else. But what it's really hard to do is to say, well, what if I lost 100 35-year-olds from my society? What's the cost of that? And that's what we need, is we need much more sophisticated levels than just comparing the health cost of A versus B or C. We need, what's the societal benefit of rescuing these patients and bringing them back to life and making them productive members of society afterwards. And that has not been done for fungal disease, never, and it needs to be.
Yes, highly country specific as well, I should think. So a question from Lilian Mukaramera. Can the Aspergillus antibody test differentiate between individuals with active infection versus people who've been exposed to Aspergillus?
Yeah, that's a very important question. So the cutoff is the key to that, right? So you can detect antibodies down to minuscule levels, and those can detect previous exposure, which of course we all have every day. So you have to determine the cutoff of the antibody test, and that's been done now in many, many studies around the world, and it varies by test. But when you've worked that cutoff out, then yes, it absolutely can tell you if a patient has got chronic ongoing Aspergillus infection with aspergilloma or chronic pulmonary aspergillosis, or in some patients ABPA or Aspergillus bronchitis, versus those who've been exposed and don't have current disease. When we did that work in Uganda, the false positive rate of antibody was 2%. So those are probably people who've had recent significant exposure and produced a strong antibody response, but don't have disease. But the positivity rate of patients with disease was exceeded 90%. So you've got a very good differentiation between active, ongoing infection and prior infection.
Okay, thank you for that. Over to you, Judy.
Okay, I think there's one question which didn't say who it was for, and I actually think both of you may be able to answer it in very different ways, but I think it's an interesting question, which is that we're facing climate change right now, and everybody's talking about it. And of course, as with temperature being a regulator of histoplasmosis, how do you think that that's affecting it, and is it affecting it, and does it affect the virulence of other fungi? So
Anita first.
Okay. All right. So I have to say I'm not. This question comes up a lot, and I think people are very interested in it for good reason. How is climate change going to affect fungal virulence? And I have to say I'm not a climate change scientist, and I feel a little bit like an imposter trying to tackle it. But there are definitely projections that climate change could expand the endemic regions for some of these pathogens like Histoplasma and Coccidioides. I think that as the temperature increases, we're performing a big selection experiment for fungi that can thrive at higher temperature, which is definitely, a requirement for pathogenesis. So I think I will leave it at that I think that, there's reason to be concerned. And there are certainly people who are studying this in detail and who are worried about what the effects are going to be for fungal pathogenesis.
So I agree with that. There is actually a paper, which I'll see if I can find while we're talking, which suggests that the virulence of histoplasma is increased with increasing environmental temperature, which is the only paper I've seen where there's a direct temperature link to a specific pathogen and what it's doing. I suspect Candida auris might have played a part in terms of its emergence, but it's also probably the fact that we spray our environment with the huge numbers of azoles, and so we get a resistant fungi coming through, and Candida auris is clearly very good at being resistant to fluconazole, does it very quickly and easily. So there may be other factors which are not just purely climate change. The one thing that bothers me more than just a slight extension of range, which we've seen with with Cryptococcus gattii, which has gone up into the Vancouver, and cocci, which is now up in Washington State, and so on, is whether we're going to get a whole-scale movement of these pathogens into whole new areas. So what if we got cocci in the sort of areas of Africa, for example, middle of Africa? What would that look like? You know, I think these endemic fungi have the potential, I think, for being translocated to whole different environments. And I think that's something that would, I think, be very significant. Hasn't been properly demonstrated yet, but I worry about it.
Okay, so I have another. I'm actually sort of a combination from two different people who both liked your talk very much, and who were asking about the physiological and shape issues. So Adnan Karim was asking if you could talk a little more about the relationship between fungal cell shape and virulence. And Kim Verberg was wondering whether the upregulation of the RIP1234 is in the conidia or in the filaments or in both. And do you think that the RIP genes and the orthologs in other pathogenic fungi can be used as a target for treatment?
Okay, so in terms of that first question, so and I can see the question in the chat, which is asking for which shape is more responsible for virulence, the yeast or the filaments. So so it's largely the yeast form that we find in the host at high temperature. And so the filamentous form is important for infection in that it generates infectious particles. And then in the host, there's conversion to the yeast form, and we see associated expression of virulence genes. So it's really the, and I maybe part of the reason, or I would say part of the reason that there's a link there is because the transcriptional circuits that drive that change in cell shape also control the expression of virulence genes. And then in terms of the question about the upregulation of the RIPs in conidia or filaments, I'm thinking that the transcription factors are upregulated in the yeast form, but I think the question might be, because you can do, I talked about a number of different transitions. So you can do a transition from conidia to yeast or from filaments to yeast. And I'm not sure if the question is asking, do you see upregulation as you transition to yeast during both of those types of developmental switches? And it's technically challenging for us to do the transition in the filament to yeast direction for various BSL-3 issues. It can be done, but so I feel like this is a little bit unsupported, but we see that. I'm going to say that my best answer is that the transition, that the upregulation occurs for both of those transitions. And in terms of targets, I think the difficulty is these are transcription factors, which are, there is very little bit in the literature out there about the potential of targeting transcription factors for antifungals, but I think the consensus is that is challenging. But perhaps my hope would be that some of the key targets of those transcription factors would be great candidates for either diagnostics or therapeutic interventions.
Okay, thank you. I'm just going to ask, I think we've just got 1 minute left if David can come up with a short answer to this one. So Nancy Keller is asking, do you get a sense if COVID-19 viral researchers and funding agencies are paying attention to fungal co-infections?
I think the short answer is yes, but people have short memories. The mucormycosis outbreak in India certainly alerted people to the fact that this was a very horrible disease, and people really were concerned about that. There is a general increase in awareness that we need to diagnose fungal disease. It's been ignored and it needs to be addressed properly. One of the other things that worries me, which I think will garner attention, is the resistance rates have been documented in southern China and Vietnam, which are up at the 80 to 90% range for azole resistance in fumigators. And I think that that's a hugely significant. If confirmed in clinical strains and so on. So I think broadly, yes.
Okay, well, I'm afraid we've run out of time now. So I would like to thank both the speakers, Anita Sil and David Denning, and also my co-chair, Judy Berman, and the MRC Centre staff who work very hard behind the scenes in helping us run these MycoTalks. So we look forward to seeing everybody again on the 16th of December, when our speakers will be Axel Brackhager from the Hans Knoll Institute in Germany and Ashraf Ibrahim from the Lundquist Institute in the US. So goodbye, everybody, and thank you very much for attending.
Very much. Bye.
Bye.
Bye.